everyone. Today's section of notes is for lesson 5.4, Evolution of Populations. So before we get started, there's just a couple words we need to define. And the first one is that a population is considered a group of individuals of the same species that interbreed. And we'll get into why that interbreed part is important. In a population, we also have a gene pool, which consists of all of the genes that are present in the population. So an example of that is this picture right here. We have a population of field mice, and we have a gene pool. In this case, the gene pool are all of the genes that these field mice have and can show in the population. Specifically, this picture is showing the gene for color. So we've got mice in this population that are black and mice that are brown. Also within that, because we know that there are dominant traits and recessive traits, the dominant trait is black, which means that we can have mice that are homozygous black, so 2 alleles for black fur, or heterozygous black, so 1 allele for black and the recessive allele for brown, as well as having some mice that have the recessive allele for brown. Over here we have the frequency of alleles, which means that the number of times we see a small b, so the allele for brown fur, or a capital B, the allele for black fur, in a population is counted up and calculated. And you can do this for any trait in any population that is related to genetics. And this is how scientists are able to actually tell that a population's genetics change, or in this situation, evolve over time. And that's what we're going to look at today. How do small populations change over time? And that is evolution of populations. So let's look at where this, first of all, this variation comes from, the fact that we have, in that case, 2 different colored mice. And there are 2 main sources of genetic variation. The first are mutations. Mutations in DNA are mistakes that can happen during, DNA replication or transcription or translation, or even during the process of making sex cells is when mutations can occur, and they lead to changes in genetics. The second type of variation is genetic shuffling, which can come from 2 different ways, and those are crossing over and sexual reproduction. So remember, crossing over is the process that we learned about that occurs in meiosis. We learned about that in our last unit. And what that means is that parts of the chromosomes literally swap places. They cross over during prophase 1 of meiosis. And this process increases the number of genotypes that can appear in an offspring. So that is one way that it causes gene shuffling, by increasing the number of genotypes that can appear in offspring. Also, sexual reproduction produces different phenotypes than what existed in the parents. If you think about that, you might have some physical traits that you don't see in your mom and you don't see in your dad, but you have them. So it's the combination of genes that now exist uniquely inside of you that gives rise to a completely different phenotype. And that is why sexual reproduction can cause shuffling or changing of genes as well. So when we look at this as an overall and how populations can change because of this variation and because of gene pools and genetic shuffling, we actually see that evolution occurs in populations through the process of natural selection. So the process that we had talked about in the beginning of that drives evolution is occurring here as well on a population scale. And natural selection affects the phenotypes in 3 different ways. And we're going to look at each one in an example, but the 3 ways are directional selection, stabilizing selection, and disruptive selection. So let's start with the first one, directional selection. If natural selection occurs in a population by directional selection, individuals at one end of the curve have higher fitness than individuals in the middle or at the other end. For example, we have birds with certain beak sizes. And in this case, the original population, which is shown by the dotted line in the graph, the average beak size in the middle was beneficial at one point in time. However, something in the population caused a change, which led to natural selection, which meant that over time, the bird's beak size actually shifted to this blue line, so that now birds with a larger beak size were living longer than birds with smaller beak sizes who were dying out. So there was a shift in one way or the other, so one direction versus the other. Now, there could have been a change in the size of food. Maybe the food evolved, the seeds got harder, so the birds with larger beaks were able to eat more seeds, so they survived longer, and the ones with smaller beaks died out. We could also see a change in the other way, which is not shown in this graph, but say the plants evolved a way to keep their seeds closer together so that there were smaller spaces that the birds could get the food out of. So those birds with smaller beaks would have survived, and we would see a graph that looked a little bit more like this green line. So if the peak of the graph is really what you're looking for, okay, you want to look at the top part of this graph, and if it moves from one spot to another on the graph, that's when you know that it's directional selection. It's moving in a direction. This is different from stabilizing selection, which is defined as when individuals near the center of the curve have higher fitness than individuals at either end of the curve. So in this case, being either extreme, either really small or really big, is not an advantage. In this case, it is an advantage to be in the middle. So the peak of our graph, our original graph, we had kind of a shallow curve, and the peak was down here. But our new graph is showing a narrowing, oops, I meant to use the highlighter on that one, is showing a narrowing of our curve, where the peak is now higher at the center. See how it's right here now instead of down here. So individuals also on either end have decreased because the extremes, in this case for birth mass, really small weight at birth versus really large weight at birth, were both causing not survival of the species, death of that organism, as opposed to having an average size or an average birth. So the graph is being stabilized to the center of the curve. And then lastly, the third type of selection in populations is called disruptive selection. So disruptive selection is when individuals at the upper and lower ends of the curve have higher fitness than the individuals near the middle. So it's the opposite of stabilizing selection. Where the original graph was this nice little normal curve, our new curve is showing a big disruption in the middle. So it peaks over here at the low end, drops drastically in the middle, peaks again at the high, and then levels off again at the end. So if you see a graph that originally had a nice little peak here, and now that peak has dropped down, that is going to be disruptive selection. Another way that species can change over time is through genetic drift. So genetic drift is not an event driven by natural selection. It is a completely random event. So a random change in allele frequency is what causes a genetic drift. And this can occur from two different ways, the bottleneck effect and the founder effect. Most often, genetic drift affects small groups of individuals when they colonize a new habitat. So if it's just a few individuals, and we'll look at a ladybug example here, and you have an example on your page as well. So just a few ladybugs from the initial population went off and decided to colonize their own populations. We have population A is now made up of mostly brown and yellow ladybugs. And then we have population B, which is now made up of mostly orange, red, and yellow ladybugs. Originally, these two subpopulations were part of the same population, but they drifted off, and who knows, maybe the population A ladybugs hopped on a log and floated downstream accidentally and ended up colonizing a new habitat. With them, they brought their specific traits and their alleles of brown and yellow with these certain patterns, leaving behind the other ladybugs who maybe in population B wandered off upstream and colonized a new habitat, and they took with them their specific traits as well of the red and the orange and the black and yellow. So genetic drift oftentimes affects these smaller populations in a much more observable way, but the random change of population can occur without some kind of driving natural force, and that is what we consider genetic drift. This is called the founder effect, specifically when allele frequencies change due to a migration of a small subgroup of a population. So population A and population B, if given enough time, may change so much that they become completely different species. And that's something that we're going to get into in our next section of the process of speciation. But this is where it starts, with just a drifting apart, and then over time enough changes will build up to cause evolution. And it all starts with the founder effect. The last thing we're going to talk about is when populations aren't evolving, there is a reason. And that is because they are in genetic equilibrium. So there's actually two men with some funny last names that developed this principle called the Hardy-Weinberg principle. And it states that allele frequencies in a population will remain constant unless one or more factors causes those frequencies to change. When the allele frequencies remain constant is when we find a population in genetic equilibrium. One of the first reasons that we need to maintain for genetic equilibrium is random mating. So random mating ensures that each individual has an equal chance of passing on its alleles to its offspring. There's no one reason why a certain male member of the species would be able to get a mate over another male member of the species. And if it is random whose mates are choosing who, then evolution will not occur. However, if there is a reason why one particular male is being chosen over another, that will drive change and that will not contribute to equilibrium. The next example is a large population. So like I said in the example about genetic drift mostly affects small populations. So the larger the population is, the more adaptable it is to random changes like bugs hopping off on a log and drifting downstream, or natural disasters that could cause change in some of the species. Larger populations are more able to cope with those random changes. The third one is that there can be no movement into or out of the population. So no immigration or emigration. Because if new individuals move into the population, they're going to bring with them their own alleles. And that is going to change the frequency of alleles in the population. So the population's gene pool must be kept separate from the gene pools of other populations. Number 4 is that there can be no mutations. If genes mutate and new alleles are introduced, then the allele frequencies are going to change. So if you end up in this picture, someone with 6 fingers enters the population and passes on that trait to their offspring, that is going to change the frequency of that particular allele in the population. So mutations cannot occur to have equilibrium. And the last one, there can be no natural selection. Since natural selection drives evolution, for a population to be in equilibrium, all the genotypes in the population must have an equal chance of survival and reproduction. No phenotype can have an advantage over the other, and that will keep the population in genetic equilibrium. But That's not necessarily the goal. This is the reason that populations don't change, but in order to survive and adapt in this world, very few populations are actually in this Hardy-Weinberg state of equilibrium. Many populations find themselves changing due to natural selection or due to mutations, and that is okay. That is a normal process, and that is how we end up with change over time, and we even end up with new species being formed. And we'll talk about that process in the next section of notes. That's all for today. Thanks for listening.